Hello everyone, I'm Virginia Mela, assistant professor in University of Malaga and also a researcher in ILIMA, Plataforma de UNAM. And today I would like to talk to you a little bit about our last publication in GUT, which has this title, Microbiota fasting-related changes ameliorate cognitive decline in obesity and boost microglial function through the gut-brain axis. As everyone knows, obesity is a multifactorial disease accompanied by many different symptoms, but in our lab, we were focused and interested in the cognitive decline accompanying the development of obesity. To understand and try to figure out the underlying mechanism of this fact, we explored by three different approaches this fact. We used preclinical studies, clinical studies, and also in vitro studies. So first, we count with this obesity cohort who underwent three different hypocaloric diets and test by different tests their improvement in cognition after the diet. And what we found was that the alternate day fasting diet was the most successful in terms of improving cognition. Then we relate the changes with different changes in the microbiota composition of these individuals. So, since obesity or this cognitive decline in obesity was really linked by many different studies with the inflammatory state of these individuals, we wanted to explore the beneficial effect of this diet in terms of inflammation. And what we found was that the alternative fasting decreased inflammatory markers in these individuals. Then, talking about inflammation and about cognitive decline, we wanted to check the main immune cells in our brain, that it's microglial cells, and how their function could be affected by this different diet. So we redirect monocyte of these individuals before and after the alternative fasting diet. And what we found was that the function of this microglial cell type improved after the alternative fasting compared with the basal condition. So, since we already showed differences in the microbiota composition of these individuals, we wanted to check if microbiota per se could be involved in somehow in microglial functionality. So therefore, we isolate the exosomes from microbiota at basal conditions, so from people living with obesity before the diet, in order to check if this microbiota could affect directly into microglial function. So using again this redirection of monocyte into microglial cell type, but this time we use normal weight people, we treat these cells with the exosomes coming from the obesity cohort. And what we found was that these exosomes from the people living with obesity impair microglial function. So then, in order to see if there is a direct link between changes in microbiota due to the diet and in cognitive decline, but also microglial function, we use this preclinical study where we transplant the microbiota from people living with obesity before and after the diet into healthy mice and check cognitive function by the novel object test recognition, neuroinflammation by different markers, and also microglial state. And what we found was that in the novel object recognition test, the cognitive performance of the recipient mice with the alternative fasting microbiota improved their cognitive performance compared with the one with the obese microbiota transplant. And this was related with an decreasing neuroinflammation in the hippocampus with interleukin 6 and interleukin 1 beta decreased, and also an improvement in morphology in the microglial cells, where microglial cells coming from the alternate day fasting recipient mice show smaller somas and longer branches. Then we also checked different markers in microglia to see if this was related with an improvement in function, and what we found was an improvement in the phagocytic capacity of these cells. That was related with a decrease in senescence marker and an increase also in cell turnover. Finally, we wanted to see if there are any metabolites responsible for that. So we study by metabolomics the different metabolite changes due to the alternative fasting in the exosomes coming from the microbiota of those individuals before and after the alternative fasting diet. And what we found was an increase in riboflavin, which was already found in another study that affect directly into microglial function. So, if you want to see more details about this paper, go to the GUT website and look for our paper. See you soon.